Today we are going to learn about peroxisomes. Is a special kind of cell organelle found in eukaryotic cells. I am Dr. Rajeev Basumatary, Assistant Professor in DR College, Golaghat. So, what is peroxisome? The peroxisomes are small subcellular compartments or a cell organelle with a fine granular matrix and is surrounded by a single biomembrane, means lipid bilayer membrane, which are located in the cytoplasm of a cell. The number and the size and the composition of peroxisomes may vary depending on cell types and different environmental conditions. So in yeast, it has seen that one yeast cell may have 20 to 25 numbers of peroxisomes within a cell. So in this diagram, the green sac or the green circles are the peroxisomes. These peroxisomes they contain enzymes that oxidize certain molecules that are normally found in a cell, mainly the fatty acids and the amino acids. The oxidation reactions of this fatty acid and the amino acids they results in production of hydrogen peroxide H2O2. So hence the name peroxisome is given to such vesicles because the hydrogen peroxide is released inside these vesicles. The peroxisome vesicles they actually function as a chamber or closed chamber that provide a safe location for oxidative metabolic reactions where hydrogen peroxide is formed. The hydrogen peroxide is potentially a toxic chemical to the cell because it has the ability to react with many other molecules within the cell. So therefore, the peroxisomes also contain certain enzymes such as catalase that can convert hydrogen peroxide to water and oxygen and thereby neutralizing its toxicity. The peroxisomes were first discovered and described by Christian René de Duve in 1960, and the term peroxism was also given by him only. Um, for this work, he was awarded Nobel Prize in Physiology and Medicine in 1974, along with Albert Claude and Georges Pallet. Now, let us look into the structure of peroxisome. The peroxisome is a spherical structure which is bound by a single layer of lipid bilayer. And inside the lipid bilayer, there is a matrix, and in the center of the matrix, there is a crystalloid core which may not be present all the time. And this is a crystalline core of urate oxidase enzyme. Unlike other cell organelles, the peroxisomes are actually created by taking in the proteins and the lipids from the cytoplasm of the cell. So they are not like the other cell organelles like mitochondria, endoplasmic reticulum, or Golgi complex or nucleus. They are not like them. So they are bound by a single membrane instead of a double membrane like unlike the most cell organelles, and the proteins which are internalized inside the peroxisome, most of these proteins they function as enzymes for various oxidation reactions. The influx of proteins and lipids inside the peroxisomes makes the peroxisomes to grow in size, and once the peroxisome is large enough, it divides through fission like binary fission, and it creates two peroxisomes. The peroxisomes are created in this manner because they do not have their own DNA to give instructions on making the protein that they need to function. Absence or reduction in number of peroxisomes may lead to a syndrome, a disease called Zellweger syndrome. The Zellweger syndrome causes copper, iron, and other substances uh, called the very long fatty acid chains to accumulate in the blood and in tissues, such as in liver, brain, and kidneys. The infants with Zellweger syndrome are often born with facial deformity and intellectual disability. Some may have impaired vision and hearing, and they may experience severe gastrointestinal bleeding or liver failure. One of the major functions of the peroxisome is the breakdown of a very long fatty acid chain through beta oxidation process. In animal cells, fatty acids are converted to medium chain fatty acid, which are subsequently shuttled to mitochondria, where they eventually are broken down to carbon dioxide and water to liberate energy or ATP. In yeast and other plant cells, this process is carried out exclusively in peroxisome only. The peroxisome also play a role in the production of bile acids that is important for the absorption of fats and the fat soluble vitamins such as vitamin A and K. The following metabolic pathways occur exclusively in mammalian peroxisomes. Number one, beta oxidation of the phytanic acid, beta oxidation of long chain polyunsaturated fatty acid, biosynthesis of plasmalogens, then conjugation of cholic acid as part of bile acid synthesis. So this was all about peroxisome and their functions. Thank you.